Hi, I'm Judy Cole, the Executive Vice President and CEO of the MIT Alumni Association, and I'm delighted to welcome you to this web production of the MIT Alumni Association.
Hi everyone, welcome to the MIT Faculty Forum Alumni Edition. I'm Barbara Moran, I'm the Managing Editor of Boston University's research website. I'm a longtime science writer and I was a Knight Fellow at MIT in 2001-2002. I'm going to serve as the moderator for this very interesting panel we have today. A reminder that we welcome your questions during the chat. Please use the form below the live stream, or you can tweet questions to hashtag MIT Better World. It's all one word. We'll get to as many questions as we can. Today we hear from 3 alumni whose research interests include Alzheimer's disease. With more than 5 million Americans currently suffering from the disease, a number that the Alzheimer's Association says might triple by 2050, Alzheimer's is said to kill more Americans than breast cancer and prostate cancer combined. It's not surprising that major research universities, foundations, and hospitals are seeking to invest record amounts in finding a cure. Promising drugs and therapies, along with some research from MIT itself, have some research speculating about an end to Alzheimer's. We'll ask our panelists about this today. We're going to start with an overview from each panelist. Each of you might want to catch us up on your career since leaving MIT and talk a little bit about your current projects involving Alzheimer's. We're going to start with, well, I'll just read each panelist's name and then we'll introduce them one at a time. We have Sarah Hopp, Matt Kaeberlein, and Anant Paravastu. Sorry, I tracked this up beforehand. I got it wrong anyway. Okay, we're going to start with you, Sarah. Can you go ahead and introduce yourself, please? And tell us a little about your research.
Yeah, so I'm Sarah Hopp. I graduated from course 9 at MIT in 2008. And after graduating, I actually went to the pharmaceutical industry for a couple of years. And I worked on drugs that kind of target the plaques part of Alzheimer's disease. And then after that, when I went to grad school, I wanted to focus on something that's a little different, because a lot of research is going into the plaques and a lot of the clinical trials haven't been going well. So I became interested in the immune system and how during aging the immune system changes and how that could lead to memory problems and then eventually have an environment permissive for Alzheimer's disease. So that's what I did my dissertation in at Ohio State. And now I'm at MGH in Brad Hyman's lab, and we study all sorts of molecular mechanisms of Alzheimer's. And I'm interested again in like the non-neuron aspects of the disease, so the immune system and molecular pathways that are present in other cells that are then kind of having an indirect effect on neuron function, maybe causing harm in aging or through other like genetic pathways and stuff like that.
Great. Okay, great. Thank you. Anant, can you introduce yourself for us, please?
Sure. I graduated in 1998 with a degree in chemical engineering, course 10. And at the time, I was actually interested in semiconductor processing. So I then went to Berkeley and did a PhD on the use of lasers to polarize nuclear spins in a semiconductor. After that, I decided to try something new, and I did a postdoc at NIH to use solid-state NMR spectroscopy for the structural biology of amyloid fibrils. So I'm not a biologist by training. The only bio I've ever taken was the intro course at MIT. I did a postdoc at NIH. I worked for 7 years as an assistant professor at Florida State, and now I'm at Georgia Tech.
Great, thank you very much. So interesting, the very different approaches to looking at the disease. Okay, Matt, can you introduce yourself to us, please?
Sure. Hi, everyone. I got my PhD from the biology department at MIT in 2002, and I did my work with Lenny Guarenti there, working on the basic mechanisms of aging in a single-celled organism, budding yeast. And since then, I've continued to pursue research on the biology of aging. I'm now a professor in the pathology department at the University of Washington. And my lab works still a little bit in yeast. We also work in C. elegans, mice, and we have a new project in pet dogs, all sort of centered around the biology of aging. And so my thinking about Alzheimer's disease really stems more from thinking about it as a disease of aging. And one of the reasons why I think it's important to consider Alzheimer's disease in this context is if you think about some of the risk factors for what causes people to develop Alzheimer's disease, aging is by far the greatest risk factor. So for example, if you're a woman, you have about a 2-fold increase in the risk of Alzheimer's disease. If you have bad genetics, you have about a 5-fold increase in the risk of developing Alzheimer's disease. Just by going from age 40 to age 80, you have about a 500-fold increase in developing Alzheimer's disease. So I think we really need to consider aging more and its relationship to causes of Alzheimer's disease. And that's sort of the way I think about it.
Okay, great. That's a bummer. I'm already halfway down that 40 to 80 path. I gotta find a way to stop it.
And I'm not necessarily advocating bad lifestyle choices, because they are real risk factors. But aging is important to consider as well.
Okay, great, thanks. I just wanted to ask one question before we go to the questions from the audience. And Sarah, since you brought this up, I'll ask it to you. Could you just give us, you mentioned about how you started your research off studying plaques. Could you just give us a very brief explanation of what plaques are in relation to Alzheimer's, and about, you also mentioned that the research in treating the plaques has run into some challenges and problems. Can you just talk about that a little bit? I think that'll be of interest to our audience.
Yeah, so basically the plaques are kind of the things that take up space in Alzheimer's. If you look at a brain section of someone with Alzheimer's disease, they'll have these big blobs of these misfolded proteins. And this forms, we think, due to like a different kind of cleavage pathway. So this amyloid precursor protein is cleaved at a variety of different places. And we think that in Alzheimer's disease, it's more prone to cleave at a bad place and then more prone to form these aggregates. And so a lot of the drug targets, because this is a very obvious pathological feature of Alzheimer's disease, a lot of drugs have been targeting maybe changing where that cleavage happens, or now some new ones are immunotherapies that are basically saying, hey, immune cells in the brain, how about you come take and destroy these plaques, like things via like passive immunization. So you give someone a shot and they then will have an immune response against this protein that's building up in their brain. And so a couple of those have actually failed, and there's a few that are still kind of coming down the pipeline. So we'll see how those end up working out. Okay.
And Anant, just to follow up, because you study the structure of the plaques, is that correct? Am I correct about that? And would this be a good time for you to show a couple slides on that? Sure. That might be a helpful overview too.
Absolutely. And in fact, when let me just share a presentation, just a few slides. So when we talk about plaques, what we usually mean is amyloid fibril deposits, although there are other things present inside the plaques. But the major component is amyloid fibrils composed of the Alzheimer's beta amyloid peptide. And that's actually exactly what my lab focuses on, the beta amyloid peptide. And so we do structural biology using solid-state NMR, and I just had to mention that we work with a team of people at Mayo Clinic and also Florida State. And so when we talk about plaques, usually what we mean is these amyloid fibrils that are sort of at the bottom bottom right of this slide. And it turns out that the assembly of beta amyloid into nanostructured materials is sort of the critical aspect of Alzheimer's disease, or maybe causative aspect. But that's actually very complicated. So there's actually a lot that goes into it. And A-beta assembles into structures that may not end up in the plaques. That may be why clinical trials have been problematic when it comes to clearance. So the assembly process is very complex. In fact, this slide looks complicated, but it's actually oversimplified. There are many, many different structures formed by A-beta. What my lab actually does is solid-state NMR. So we look at isotopically labeled versions of beta amyloid. And I'm not going to get into the details of NMR, but we obsess about how every atom is organized in 3 dimensions. And in terms of where the field is at in terms of structural biology, we have a pretty good idea of how the fibrils are composed, what they look like on the molecular level. But there's this growing belief that fibrils themselves, by the time they deposit into the plaques, are not the major problem. So the current research is on oligomeric states, which are much smaller than the fibrils. and probably are more mobile, diffuse into tissues, interact with cells more effectively. And that's what we're studying right now. So we're placing isotopic labels and trying to see how all the molecules fit together. If we can understand the assembly pathway on the molecular level, we think we can actually, design therapeutics. And I would add one more thing, and that is that I don't think therapeutics is everything. I think detection is also extremely important. And so if you can detect if you know what to look for in terms of a specific toxic structure, and early detection of the disease, I think, is also an extremely important thing to work on. But I'll let others speak on that. In fact, I think I'll learn a lot during this discussion.
Let me, Sarah wanted to say something about the fibrils and pox, and I think that's relevant here. And then Matt, you can talk about the immune system aspect of this. That's okay. And then we'll go to questions from the audience. Go ahead, Sarah.
Yeah, so one of the things that we study in the lab that I work in now is the toxicity of the fibrils and the oligomers, and how that's basically one of the ways that they're acting is actually on neurons. As neurons are sending out like processes to communicate with other neurons, there's these synapses that form, and those oligomers actually cause dysregulation of ions. So like calcium ions are how neurons communicate with each other, and when there's too much of them, it causes problems. And one of the pathways that we study that's triggered by this calcium overload is the calcineurin pathway, and my lab's been studying that for about 5 years. And there's some interesting potential drug targets in that pathway as well that haven't really been looked at by like the pharmaceutical industry, but we're really interested in, because we've seen that this is one of the ways that the oligomers are actually causing harm, is making the neurons lose these connections to other neurons, and we can like track that with fluorescent labels and neurons and stuff like that.
Right, just a much finer tuned understanding from what we've heard before, really. Matt, anything you want to add to that?
Yeah, sure. So I want to come back initially to the point that Anant made about detection, because I think that's a really important component here. I think the earlier that we can detect individuals who are going down the path towards Alzheimer's disease, the easier it's going to be to actually intervene and in the disease process and slow it down or stop it. So I think that's critically important. The other point I wanted to make comes back to these immune therapies, and I think this is a really good example of where a very promising strategy falls down when you don't consider the component of aging in the whole equation. I think, as probably everybody realizes, our immune systems stop functioning as well as we get older. That's why an elderly person doesn't respond to a flu vaccine as well as a young person. And so these strategies that rely on our immune system to actually help fight off the disease are much less effective in an old individual. The problem is most of the basic research in this area uses young mice for these studies, and you lose that aspect of the aging process itself and how that affects therapies. And so I think we really need to get to understanding how the aged organism is going to respond to these therapies before we really can predict whether they're going to be effective in people.
Very interesting. Great. All right, let's go to some questions. The first couple questions from the audience here about lifestyle. So let me just go to the first one here. While the medical pharma industry is looking for a drug that stops or cures a disease, are there lifestyle nutritional change protocols that are showing results to reverse early dementia? And do they seem to reverse if it's interrupted? Any comments on there? Matt, you want to start with that? You're nodding.
Yeah, sure. So, I mean, I think it's, you always want to be careful about drawing too strong of conclusions from these sort of epidemiological studies or correlative studies. But I think most people would agree, and I certainly believe, that healthy lifestyle can play a big role in protecting yourself against age-related declines in cognitive function, probably also most cases of Alzheimer's disease, although I think what we call Alzheimer's disease is not always one disease, and so I won't say always, but I think in general, as you might expect, eating a healthy diet, exercising regularly, not over-consuming alcohol, all of those things probably confer some level of protection against these losses of cognitive function and onset of dementia that go along with old age.
Okay. Anybody else, Sarah or Anant? Do you want to add anything to that?
Yeah, there was an interesting study it was a very small study that came out not too long ago from some researchers at UCLA that did a diet intervention that seemed to have some modest effect, but that's the only one that's kind of been like a clinical trial style thing on like nutritional supplements and lifestyle changes. And it was very, very aggressive, but it actually did show memory improvement, which is one of the first studies.
Right.
What does diet intervention mean?
Basically, I read the paper, and it's a giant spreadsheet tailored to someone's blood tests that gives them all of the nutritional supplements that bring them up to some standard that they're looking for.
So it's like a nutritional supplement. It's not like change your diet to a low-fat or high-fat diet or something.
Like basically changing to a vegan diet on top of all the supplements. It's really an aggressive study, and it's very small, but it was interesting.
Yeah.
I'll also add one thing because I think you brought up this idea of reversing the declines in function. I think that's a few years ago I would have been very skeptical of that idea, but I think that there's actually reason to be optimistic that at least up to a certain point there is the potential to actually regain or reverse some of these declines in function that go along with the aging process. And so some of this has to do with rejuvenation of aged stem cells and the environment that they're in. But I actually don't view that as a lost cause. I sort of feel like there probably is a point in the Alzheimer's disease process where it probably becomes too late for reversal. But I think that early on, there actually is the potential to actually rejuvenate functionally some of these aspects of dementia and Alzheimer's disease.
Wow. Okay, that's good news. I like that. There's a question from Doug in Illinois. How are we closer to being able to definitively diagnose the disease while the patient is alive? And Anant, this goes to a little bit about what you were saying before about detection. Can you give me a sort of sense of where we are, or the global where we are with sort of detecting, and how early, and where is that all headed?
So first I want to say I'm not a doctor. Yes. I'm a chemical engineer. One should always take what I say with a grain of salt about anything, but especially this. So my understanding is that there is no diagnosis for Alzheimer's disease other than taking a section of your brain, which is not advisable while you are alive. And so, to actually identify Alzheimer's disease, what you'd be looking for is plaques in the brain. And I believe that is only done post-mortem. So technically, a doctor may believe that a patient has Alzheimer's based on the symptoms. You can actually look for A-beta levels in spinal fluid and in blood, but I don't think any of those things are really considered a definitive diagnosis. I also don't believe that anything you can measure has been highly correlated with the actual disease. For example, you can measure plaque levels in a brain scan, but I don't think that's highly correlated to any known symptoms.
Okay.
How about if we expand the definition to Alzheimer's-like dementias, though? Because I think you're right in the sense that you're giving sort of a very molecular definition. But I think if we expand this to be a little more broad, then there certainly are clinical diagnoses that can be made for Alzheimer's-like dementias, and the extent to which these are identical diseases, may not really make that much difference in the long run.
Mm-hmm. Mm-hmm. Sarah, did you want to add something about PET trials,
PET
scan trials?
Yeah. So my lab is like probably 50% a clinical lab. Actually, I'm not that kind of doctor, but a lot of the people I work with are. So we get a lot of brains that come in post-mortem that we do work up on pathologically. And so we get a lot of cases where someone comes in where we think they have Alzheimer's and they don't have Alzheimer's on that molecular level. So how relevant is that if they looked like they had Alzheimer's to a trained doctor? But we also have some cool stuff at MGH where there's these PET scans that can look at the plaques, but there's also PET scans that can now look at the tangles, which is kind of interesting because those are inside of neurons and maybe are more correlated with kind of the staging of Alzheimer's disease, and you might be able to catch that earlier. And then there's also some new PET scans for looking at immune changes in the brain, which also seems to be really correlated with things like cognitive test scores, perhaps even better than the plaques were. So maybe that could be another great way to kind of get an early diagnosis and maybe have an early intervention possible.
Anybody else want to add anything about that?
No, I guess I'll just add, briefly that I think these advanced imaging methods have a lot of promise, and I know that's a big emphasis here at our Alzheimer's Disease Research Center. The director of that center, Tom Grabowski, is doing work on studying changes in brain function using these imaging methods and using that as a predictor for either early mild cognitive impairment or progression to Alzheimer's disease. And again, I think the earlier that we can diagnose these things, the better the opportunity to actually intervene and either slow it down or halt the process.
So along those lines, let me ask you this question from Boca Raton about what medications are available today and are they effective? And right, so intervention is if we can detect how do we intervene? Just tell me, can you overview of what's available and how effective are the stuff? I know you guys are not clinicians, we've established that, but
well, I'll start and I'll be a little bit provocative. So the answer is I don't know of anything that's really effective. There might be something out there, but I'm not aware of anything. And in talking to people who treat patients, that's sort of the impression that I've gotten. So there's a pathway that we work on in my lab called the TOR pathway. And there's a drug that targets that pathway called rapamycin. Its clinical name is sirolimus, or Rapamune is the generic version. So preclinically, in every Alzheimer's disease model mouse model that exists, rapamycin has been shown to both decrease the amount of A beta or tau and also improve cognitive function. And as far as I can tell, nobody is actually doing a clinical trial for Alzheimer's disease for rapamycin, which I think is unfortunate, and I don't quite understand why that's the case. I think there's a pretty good chance that rapamycin would be effective at slowing the progression of Alzheimer's disease and perhaps even reversing some of the cognitive declines that go along with Alzheimer's disease. So again, I think the preclinical data for rapamycin is about the best of any drug that I know about. It's FDA approved, it's available as a generic, and I don't understand why the Alzheimer's field has not tested rapamycin yet in clinical studies.
Interesting.
So I can add something about effective drugs. I have read that there is actually two, or at least classes of drugs that are effective, but being effective is not enough. So smoking is actually Alzheimer's protective. So I believe nicotine, there is evidence at least that it's Alzheimer's protective, but I think you know what I'm going to say next, and that is that's not necessarily the best idea, right? I've also heard that long-term usage of NSAID drugs, non-steroidal anti-inflammatory drugs, also has been shown to have anti-Alzheimer's properties, but it promotes the risk of stroke. So those are interesting research angles, right? Because there is some evidence that they do protect, but of course they have other problems.
Yeah. Sarah, you look like you want to say something about the NSAIDs. Do you have yeah, go ahead.
The NSAIDs are kind of my favorite because that's kind of what got everyone into studying the immune system in Alzheimer's disease, because there are these large studies showing that that people with arthritis for some reason were less likely to get Alzheimer's disease. But it turns out when you actually do an interventional trial with NSAIDs, it's not helpful. And some of that might be by the time someone has the symptoms of Alzheimer's, it's too late to intervene with the drug. So maybe if you have aches and pains, don't worry about taking ibuprofen because it might help you not get Alzheimer's disease.
Yeah. We have a couple more questions here, more specifically about diet, which are interesting. First of all, one caller, Beth from Idaho, is calling us on saying that we shouldn't characterize a vegan diet as extreme. People are on it for lots of reasons. So just want to, get her out there. And let's see, where is this?
So can I make a quick comment on diet?
Absolutely.
I think, one of the reasons why diet could be important is because we know that if you have diabetes, that increases your risk of Alzheimer's disease by about twofold, maybe a little bit less than that. So people who eat a healthy diet or a vegan diet are probably at lower risk of developing diabetes. They have better glucose homeostasis, and so that certainly could contribute to a decreased risk of Alzheimer's disease and other dementias.
Could you talk a little bit more about glucose? There's a few specific questions about glucose and the link between Alzheimer's and diabetes. And can you tell me what's going on with that?
That's outside my area of expertise. I could certainly speculate a little bit, but I don't really. So if either one of the other panelists know the answer to that more definitively, I'll defer to them.
I don't know any much definitive stuff. I know. Oh man, it's getting loud in my background. Sorry about that. That one of my colleagues from graduate school was studying insulin in the brain as an intervention for age-associated cognitive deficits. But the thing is, insulin acts totally differently in the brain than it does in the rest of the body. So the connection, sometimes it's hard for me to make the leap from one to the other. So that's all I have.
Okay.
I think. Go ahead.
I'm sorry. I can comment on the molecular level behavior, but I think this is not really established to be true. But insulin and another diabetes-related protein called amylin are known to form amyloid fibrils. And as we mentioned, A-beta amyloid fibrils are associated with Alzheimer's disease. And at least in the laboratory, there is some evidence that an amyloid of one protein can actually promote the formation of an amyloid in another protein. And so there are those who say that there's actually a molecular level connection between different amyloid diseases, and insulin is one of them.
Yeah. Okay.
Okay. I also will just add, I think that, it also probably goes beyond the molecular sort of explanation at that level. I think that there's a lot to systemic inflammatory processes.
Yeah.
And we know that diabetes is sort of associated with systemic inflammation, and that probably contributes to a lot of the changes that are happening in the brain at both the metabolic level and also the way that the cells are signaling to each other. So I think there is this systemic component that goes beyond the sort of A-beta and amyloid interactions in the brain itself. So I think it's all probably tied together at some level.
The inflammation connection is something I'm just starting to hear a lot more about. And so Sarah, do you want to comment on that, or Anant, about how is that sort of emerging as a more studied factor in Alzheimer's?
I mean, so there's a lot of changes that happen with the way the immune system functions in aging. And since aging is the largest risk factor for Alzheimer's disease, sorry, I'm getting an echo in this part.
Aaron, why don't you take it and let it.
Well, this is way outside of my sort of understanding. So inflammation, is I know that in general, it's related to an immune response, and I think that there are other terms. In fact, I think I'm actually trying to set up Matt to answer this question because I can name a few other things that I know to be related to the question, but I don't really know the answer. So there's this question of clearance, which I'm not sure is related to the immune response. And then there's also this question of oxidative stress. Maybe I should just stop talking, but I do not really know the answer to that question.
Yeah, so I'll comment on it from the aging perspective. So one of the things that we know happens as our bodies get older is we accumulate senescent cells, and these senescent cells give off pro-inflammatory factors. So they don't die, they kind of stay in our body and they secrete these pro-inflammatory factors, which then leads to a systemic inflammatory state. It gets into the circulatory system and induces a systemic inflammatory state. And so there's accumulating evidence that if we can actually shut down this secretory profile that senescent cells give off, that can actually have functional improvements in the aged organism. And you may have heard about this idea of treating Alzheimer's patients with blood from young people. There's actually a company that's trying this. I'm not advocating that at all, but that stems from this idea that there are these systemic circulatory factors released by senescent cells that are not present in young people, that are present in old people, and that it may be possible to kind of dilute out these factors and improve outcomes in elderly people, including cognitive function. So that's part of where this systemic inflammatory environment comes from, is these senescent cells.
I can hop in and talk a little bit more now that my sound issue is a little more calmed down.
Thank you.
So I study the immune cells in the brain, which are called microglia, and so they also get affected by the peripheral immune system, despite being kind of trapped in the bubble that is the brain behind the blood-brain barrier. So what happens when they're in an environment where there's a prolonged level of this inflammation is that they actually get worse at doing their normal functions. And one of their normal functions is garbage collection. And as you imagine, in a disease where there's buildup of plaques, that garbage collection is really important. And so it also turns out that the plaques themselves cause inflammation to those immune cells that are in the brain that then turns off their ability to clean up the plaques. So it's kind of a toxic feedback cycle that is just no good. And that, combined with the dysfunction in the peripheral immune system and aging, probably contributes heavily to Alzheimer's.
Mm-hmm. Sort of backing up a little bit to the sort of big picture diet question, because you've talked a little bit about diet, and it's been like, follow a good diet, but there's specific questions about sugar, fat, alcohol consumption. Are there more specific guidelines about what people should do, or is it still all just, fruits and vegetables, healthy diet,
Yeah, I mean, I don't know that we can really be a lot more definitive than that. I think, the nutritional science is still working out, what exactly is the optimal diet. And I think the idea that there's going to be one optimal diet for everybody is a little bit too simplistic. We know that different people respond to diet differently. So I think we don't know. I will say, in the aging field, what seems to be an emerging paradigm is, at least for people that are under about the age of 65, lower protein is generally better than high protein. And there's a biological switch right around the age of 65 where people who are eating a high protein diet actually, at least for mortality, so for survival, seem to do better. But again, it's really early, and I'm not, that's sort of the paradigm that people are thinking about now, and that's new in the last few years. And I don't subscribe to that sort of diet, because I'm not sure whether 5 years from now we're going to be someplace else. So I think for now we're kind of still stuck with, eat healthy, fruits and vegetables, not a lot of simple carbohydrates, and probably most important is not too many calories, right? So so I think that's probably the most obvious thing that almost everybody would agree with, that eating too much in general is not a good thing. But I don't think, at least I'm not comfortable enough to make recommendations beyond sort of what we all already know about what eating healthy means.
Okay. I'll ask one more diet question, and I'll stop, just because this one is so interesting, and I don't even know what to make of it, but I'll see if you guys do. This is from Rich in Arizona. He says, I read that Alzheimer's per capita in India is much less than in the United States. Association with red meat and turmeric consumption has been proposed. Any thoughts on that? The turmeric, we knew there had to be something. Yeah.
I told you
come out.
it was going to come up.
You said cumin. You said cumin.
Yeah, well, it's curcumin, which is part of turmeric, I guess.
Okay, so what is the list?
These population studies kind of had us looking at India, because they don't get Alzheimer's as much, and saying, okay, what's different there? And this curcumin is in turmeric, and it's a potent antioxidant. And there's just been some interesting research, mostly in, again, mouse models of both transgenic Alzheimer's as well as in aging models, and it seems to be really promising. The thing is, the amount of turmeric that you have to give a mouse is really, really high. So imagine you had to eat pounds of this stuff to get the correct amount of this curcumin.
Yeah. All right.
So I have a couple comments about that. One is that one has to be very skeptical with statements like that. For one thing, the curcumin story has been criticized because it may be possible that Alzheimer's is just underreported in India. So I'm not sure we really have good statistics to support the idea that Indians get Alzheimer's less. I'm Indian and I have family members with Alzheimer's. I don't know what that means. But a couple things on the molecular level. So curcumin is actually very similar in structure to Congo red, which was one of the original dyes that actually was used to identify Alzheimer's plaques. And it is also true that curcumin is able to cross the blood-brain barrier, which is really important for an Alzheimer's drug. So it has certain properties that I think are encouraging, but I'm not sure exactly whether or not curcumin is really the key to avoiding Alzheimer's.
Yeah, that raises an interesting point, though. So some of these amyloid binding dyes in invertebrate models have been shown. So if you take, say, the A beta gene and you express the protein in invertebrates, you can cause aggregation and toxicity. And some of these amyloid binding dyes have been shown to prevent that. So there is a plausible biological mechanism by which curcumin or some of these other amyloid binding molecules could get in there and kind of disrupt that aggregation process. So I agree, we need to be a little bit skeptical about these epidemiological links for the reasons that are not mentioned, but I also think there is some reason to think that biologically it could make sense.
Yeah. Well, that's what epi studies do, right? Give you little clues, right? About,
they give you hints. That's
hints. Right. Question from Bert in Denver asking, is there a aluminum link to AD? Should we avoid cooking with aluminum cookware? Matt's giving me that look.
I don't know. I'm giving you the I don't know look.
Yeah.
My dissertation advisor wrote his dissertation on this in like 1980.
Right. It seems like I've been hearing about this for like 20 years, right?
And I think that the result is, well, if you inject aluminum into something, that's bad, but probably eating with it is okay.
All right. Okay.
So I use aluminum cookware.
Not okay, and you're okay so far. It's because you're putting all that turmeric on everything. That's
true.
Okay, so this is a interesting, provocative question from Tom in Sudbury. It says, I won't hold you to the title of this program. The title of the program, talking about the end of Alzheimer's here. I won't hold you to the title of the program, but it'd be useful to hear some wild guesses from panelists of a date when Alzheimer's will be licked. When we predict a cure for Alzheimer's, are we talking never, 100 years, 50 years?
Yeah.
Any?
So I'll start, and I'll be, again, a little bit provocative. I think it's a mistake to talk about curing things like cancer or Alzheimer's disease or heart disease. I think a better approach would be to try to understand the mechanisms that lead to these diseases. And again, my bias is that we really need to understand the mechanisms of aging and to try to delay those mechanisms or slow them down to the point where we push back all of these diseases of aging at the same time and expand health span, because that's really what we're all interested in. We're interested in the period of life that's spent in good health, free from these diseases. So we really should be talking about maximizing health span than trying to cure diseases. I think it's possible to cure diseases at the individual level, so we can cure an individual person's cancer, but at the sort of population level, I think it's a mistake to talk about curing these age-related diseases.
Okay. Anant or Sarah, do you want to add anything to that?
Well, so I would add that whenever somebody asks me for a number, whenever I ask somebody for a number and the answer does not have a number in it, I get kind of aggravated. So because of that, I'm going to give an answer of 20 years.
Oh,
that is based on almost.
That's good. That's an optimistic, can-do MIT answer. 20 years.
I would also guess like 25 years. I think that that's about how long it's going to be for some of these drugs that are looking promising in early preclinical stages to get through the pipeline. And I think that once we get a couple through, we're going to get a bunch through as we start to understand stuff more.
And I'll just be the contrarian and make the point that the war on cancer is more than 25 years old.
Excellent.
But so is the war on Alzheimer's.
What do you think? Let's talk about drug targets again a little bit. Sorry, I'm getting a little feedback here. Where do you think are the most promising drug targets now for Alzheimer's, sort of way back in the pipeline? And do you think that there are? What do you think what do you think's going? Because there's been a lot of very big disappointments in Alzheimer's drugs over the last couple of years. And are people starting to sort of rethink entirely where they're looking at targets, and where are some places? I know, Matt, you talked about this a little bit before, but do you guys have anything else to add about drug targets?
Well, I'll go back to my theme of the hallmarks of aging as good targets. So I think rapamycin, I already talked about the mTOR pathway. I think there are a series of people who are developing drugs that target senescent cells. They're called senolytics. I think those have the potential to impact that systemic inflammatory environment that I talked about, as well as immune senescence. So those, I think, are two exciting candidates in the field of geroscience or aging research that could have a pretty significant impact on Alzheimer's disease. And again, I think maybe the cure isn't necessarily the right way to think about it, but if we can push it back to the point where people are not experiencing the significant declines in cognitive function and dementia, push those molecular processes back. It's kind of like, the strategy of treating prostate cancer, right? If it's not going to kill you, you don't have to worry about it. And so I think if we can push the functional declines back to the point where people aren't experiencing that at the end of life, that's probably enough.
Right.
So I think those are a couple of good candidates from my area of research.
Right. Anant, want to add anything?
Well, so I wonder if the answer is you need more than one drug. So most of the drug targets that I have seen have to do with plaque clearance, and I think the disappointments have suggested that plaque clearance is not enough, but I suspect that it's still important. So I think, as we look at new mechanisms and new targets, I think there's probably, I think the answer will be probably more than one type of drug. I'm not sure, though.
Okay, great. We have about a few minutes left, so I'm going to ask one last question that came in from Alice in New Haven, since this is an MIT forum. She asks, what MIT-specific research excites you in this field now? What do you know about coming out of MIT that you feel like is exciting for the future of Alzheimer's research?
I think that Li Wei Tsai in course 9 is doing some really interesting work on some immune pathways that are relevant to this, and doing some interesting genetic work to try to figure out some pathways that could maybe lead to is really early kind of work that could lead to potential therapeutic targets, and that's pretty exciting.
Great. Matt and Anant, do you want to add anything from your alma mater? No.
Well, this isn't exactly an answer to the question, but I do want to mention Susan Lindquist, who recently passed away. So I guess we can't say that her research is current, but it is very recent, and I think it has, been extremely influential in the field of amyloid diseases. So she's worked on prions in yeast. And so that's my answer to the question, even though it's not an answer to the question.
Great. Good. Okay, wonderful. Thank you. That was a great session. We're going to wrap it up now. On behalf of the Alumni Association, thank you for tuning into this Faculty Forum Online Alumni Edition. Thanks again to our alumni panelists from MGH, Georgia Tech, and Washington. We didn't get to all the questions, but we'll forward all the questions to the panel. You can tweet about today's chat using the hashtag MIT Better World, and send follow-up questions or feedback to alumnilearn@mit.edu. Thank you again for watching, and thank you to all the panelists. That was a great afternoon.
Thank you.
Thanks again for joining us. For more information on future MIT Alumni Association productions, please visit our website.